Hello there, and welcome to Preprints in Motion, a podcast taking a deep dive into the fast-paced world of preprints.
Join us as we sit down with early career researchers, discuss their latest preprint, and find out about their journey through the muddy marshes of academia.
Hit that subscribe button, leave a rating, and find us on Twitter at MotionPod. Support us by heading over to buymeacoffee.com/preprints. But for now, let's get into the show.
There's a rough outline of questions, but
Okay,
I think Emma was a bit scant because, macrophage metabolism and heterogeneity, that's my wheelhouse. So this is
All right.
finally an episode for me.
Oh, sweet.
I assume other people listening will also enjoy it, but I'll enjoy it. I will definitely.
Hopefully.
So let's start with, who are you? Where are you? What do you do?
Well, I don't know. Academically, I guess I'm a postdoc, currently in Leiden in the Netherlands, at the University Medical Center there. But yeah, that's only been the past 2 years. I mean, I was born and raised in Vancouver, which is where I did my bachelor's and started my PhD. And then through just, random chance, ended up in Glasgow. My supervisor ended up taking a position there halfway through my PhD. But yeah, I was lucky enough to have the opportunity to join her over there. And then, ended up here after finishing the PhD there.
There is a slight Scottish twang to the accent.
Yeah, I think it's gone away since I've been in the Netherlands. It definitely comes back when I talk to people from the UK, but... for sure, when I started here, the whole department was confused. They were told a Canadian was coming. And then, after hearing me, just assumed I was Scottish or some form of British.
Yeah. So, living in Scotland for any duration of time, you're going to drink a lot of whiskey.
Aye.
Best whiskey. What should everyone listening go out and buy?
Oh, no, whatever you like. I don't know. It's, I don't have a refined palate. I like them all. I have a few favorites. I like the smoky. It's the Isla whiskies, of course. Who doesn't? Yeah. I think any real whiskey lover tends to lean that direction. But yeah, I have kind of an arsenal that I usually go for. The Oban 14, which is not a smoky one, is one that I really enjoy. Yeah, if I have some spare cash, then I shell out for the Lagavulin 16, which is I think almost mainstream now after Parks and Recreation made it famous. But
I'm drinking a whiskey sour tonight as it happens, so it's very appropriate.
Actually, I brought my whiskey love to Leiden. I think we've got a good group of people here that we're very keen to try and learn to love whiskey. But we actually started making old fashions, which I had to say is just if you have to mix whiskey into a cocktail, it's the best one because it's
a good way to go.
Pretty much just straight whiskey, but with a bit of flavor.
Okay, so we're a science podcast, so we should talk about science a little bit. Smoky, though, I'm all about the smoky. A good Talisker if you want something relatively cheap. Yeah. But anyway, science. So macrophage heterogeneity, all about that. I love that. Right. My entire 4-year PhD was on macrophage heterogeneity.
Well, you know more than me then.
Well, it was in fruit flies, so almost certainly not, unless you want to talk about flies. So could you give us a little summary of what this preprint is all about, what you did?
Honestly, just we kind of copied what other people had done, but just applied the technique to something a little different. So, I mean, I guess one of my... my whole PhD was focused on immunometabolism and on T cells mostly. So I've been in the field for a while. And I think one thing that always kind of lacking, in my opinion, was a real drive to understand just what is happening metabolically in immune cells and in their normal state. And then, of course, my interest is infection, so how that changes during infection. And I think people so quickly just jumped to transgenic models, using in vitro systems, knocking stuff out, using inhibitors, but not actually trying to understand, before you do any of that, what does the metabolism actually look like? And I think now we're actually now getting to that point as technology gets better at addressing these questions. So there were about 3 papers that all came out roughly at the same time that were the first to show that or apply flow cytometry or mass cytometry to look at metabolic targets, enzymes and transporters instead of immune markers, which is what you would normally do. So it's pretty straightforward thing. And I think the only reason that it hasn't really been done so much before is because the companies that make these antibodies, haven't been interested in metabolism. Yeah, with following on that, we found ways to incorporate this into flow. Others, as I said, others did it first. And the other papers focused really on human PBMCs, stimulating in vitro, this kind of thing. A really elegant paper looking at using the same technology, but to look at how CAR T cells respond in vivo in mice. But yeah, we just decided, well, why don't we just apply this to tissue macrophages? Because, they're an incredibly diverse population of cells. Yeah, the way they behave and act is different in every tissue. And there's a real, interest amongst immunologists in understanding this in more depth.
Yeah, I mean, the metabolism field, it was nothing for years, and then suddenly kind of Luke O'Neill, I think, probably led the charge a bit, but it's just suddenly loads of metabolism stuff, especially in macrophages.
Yeah, it's to be honest, for a while I thought it was a fad that would eventually die off, but it's still going strong, very strong. And I mean, ultimately, once you realize that metabolism is really just what drives everything in a cell, it's hard to get away from.
Just purely selfish question here, but how well do those antibodies work for flow? I mean, presumably well, because you've done it. But they seem to work very well in our hands. I mean, we're putting a certain amount of faith in the manufacturers or the suppliers, because at least one of the companies that we order through, several of the antibodies are knockout validated in their hands. So yeah, we have some faith that that they work as they should. But I mean, if you're just talking about pure signal, we get great signal. And of course we see changes depending on how we stimulate the cells or where the cells come from. They seem to work very well. Good, because I am. I'm on the neutrophil project at the moment, but we've got some interesting metabolism data that we could do with verifying other than qPCR, and we can't do seahorse. So. But yeah, anyway, my selfishness aside.
Yeah, no, not selfish at all. I mean, I think it's something that if anyone wants to try this, will want to know.
So could you give us a little bit of an overview of what mass cytometry is and what spectral flow cytometry is? Because I think most people at least, will probably be familiar with standard flow cytometry, but these are two relatively new, I think, techniques.
Yeah, well, mass cytometry has been around for, I think, a while now, but instead of using lasers and excitation and emission of the light or the spectrum, you just conjugate a heavy metal, and then it goes through a mass spec basically, and then if that metal is present on the cell, then you get a signal.
I mean, it basically lets you do loads of targets, right, at once. It's more than you could do with standard flow.
Yeah, I think ultimately, from what I can tell, because there's a lot of mass cytometry done, cytoph done in our department, and it still does seem to be the best in terms of maximizing the number of markers you can look at once, and also in terms of the resolution. I mean, anyone who does flow cytometry will understand the pains of compensation, especially as panels get bigger.
It's a bane of my life.
And that's really, from what I understand, less of an issue with cytoph.
I spend most of my time doing flow panels, and it's mainly just compensating.
Yeah,
and then
exactly.
realizing the panel doesn't work, so you got to start again.
Yeah.
I hate it.
Yeah, I have to say, so spectral flow cytometry, which we're doing now, is a bit of a godsend in a way. I mean, it certainly has its kinks, or it's its fair bit of troubleshooting. But the way it works is, instead of looking at just one peak emission, it takes the entire spectrum of that particular cell or target, and so based on that, it's called spectral unmixing. So you record the single stains, it knows what that spectra looks like, and then in a mixed sample you have all these various spectra. Somehow the machine magically knows where it comes from, and then as long as you design your panel well, you don't have to compensate at all. You can make minor adjustments if you need to, but it honestly just, the biggest thing is it saves so much time on the cytometer, because you just go and you run your samples, and then there's not much you can do compensation wise. Yeah, that's my terrible explanation of cytoph and spectral flow cytometry.
We've got one in the department, but I just haven't been trained on it yet. We had one where I last worked, and I hadn't been trained on that either. It's on my list to do, but I just can't find a good enough reason to do it at the moment.
Yeah, just save yourself time.
I know. So maybe we should talk a little bit about macrophage heterogeneity, because I assume most of our listeners are not immunologists. So I mean, you've looked at macrophages in different tissues, so could you explain why that's important to do?
Yeah, in general, I think immunology, has to start to looking in tissues because that's where all the immunology really happens. And macrophages are really key in any immunological process, I think. Yeah, I mean... I have to say that my bit of a disclaimer here is my entire PhD was on T cells, so the macrophage side thing still feels relatively fresh despite working on it for 2 years. But there's all sorts of disease phenotypes with macrophages. They really are key orchestrators of several physiological processes, from tissue organization to neurotransmission to you name it. And of course, every macrophage in every tissue is going to be specialized for whatever function it needs to do. So understanding this heterogeneity, of course, I guess helps you get a better picture of where macrophages need to function and how.
I mean, that's why macrophages are just the best cell, and there's no argument against that, because they do, like the macrophages you have in your heart versus the ones you have in your lungs are very, very different cells, despite being called macrophages, and they're involved in everything you can possibly imagine. Macrophages seem to be having a role in it. So they're the coolest cells ever to work on. It's interesting that you did T cells to macrophages, because I went the other way and did macrophages to T cells to now neutrophils and T cells. So I know macrophages, not so much on the T cells. And I skipped I went CD8s and then CD4s. So I've yet to build up any kind of working knowledge of T cells.
Yeah, I mean, I think, it's like your first love, right? I don't know. T cells are always, I think, number one. Macrophages, of course, they're getting up there. But yeah, I think it's probably proportionate to just how much time you spend on it.
Yeah.
But yeah, it's maybe working its way to a tie.
I spent a lot of nights sat alone with macrophages. So, it's a bonding experience, if nothing else.
Yeah, I don't know. It's my biggest problem with tissue macrophages is just they're such a pain in the ass sometimes. I miss taking out a lymph node or a spleen and just mashing it, and then you're ready to go.
Try working on neutrophils. They are so much worse.
I can imagine.
They're awful. Horrible cells to work with. I mean, they're great if anyone I work with listens, but they're awful. They just don't do anything. Well, they do a lot of things. They don't do anything you want them to do. And they're a pain to work with, technically, as well. So macrophages metabolism was one of those things. I kind of mentioned Luke O'Neill. I think if anyone knows anything about macrophage metabolism, his name's probably involved in that. He gives a really good talk, if you can keep up with his incredible talking speed. He needs to be on half speed. If you can record him and watch about half speed, then it'll be a good talk. Also, a really nice man, actually, as it happens.
Yeah. I mean, I've not met him personally, but I get that vibe.
I've had a drink with him. I've had a drink with him once. He's in a band. I mean, so he's cool.
I've seen him play at one of my conferences, yeah.
Oh, is he good? I've never seen him play. Is he good?
Yeah.
He doesn't listen. I'm almost certain he doesn't listen, so it's okay.
Yeah, I mean, it was a long time ago, but I remember, the energy was flowing for sure.
So could you explain how metabolism works in macrophages? Because it's different between a macrophage that is active versus not active. It's also different between the subsets of M1, M2 macrophages. Talk a little bit about that.
Yeah, well, the whole M1, M2 paradigm is, I think, something that the field is very actively trying to abandon.
That's another question. That's my next question.
Yeah, but my current supervisor, Bart Everts, was, involved in some of the seminal work on that. But, the initial findings, I guess, which I think actually date back well before the field became what it is today, was that if you stimulate macrophages, traditionally in a dish with LPS or anything that's pro-inflammatory or will induce pro-inflammatory cytokine production, really strongly associated with this increase in glycolysis. So they just take up loads of glucose, and secrete loads of lactate. Yeah, and then on the other side, which I think was figured out to more recently, well, relatively speaking, is that if you polarize macrophages towards what we'd say more of a wound healing or regulatory phenotype, something that would, in theory, dampen down the immune system rather than promote inflammation, and more is involved in resolution, really instead switch towards other pathways. So they use more lipids and more amino acids, so particularly glutamine. Those were the initial in vitro findings, M1, M2. I think it's as we get more into the mechanisms of this, of course, it's we're learning that it's much more complicated. I mean, if you look at some of the M2 papers, I mean, they clearly use loads of glycolysis as well, or loads of glucose.
So why would a macrophage that's been activated want to switch its metabolism?
Oh, yeah, it's a good question. At least from my work with T cells, one of the hypotheses I always liked was that there's a combination of just competition between pathogens, as well as just an adaptation to kind of global metabolic changes. Like you get sick, you end up with hyperglycemia, and then that gives you fuel to to act as a T cell. So yeah, I don't know if it's the same thing with macrophages. I mean, if you get some sort of inflammation, you get trigger, and then there's more metabolites coming in, whether it's a consequence of other metabolic pathways. One of the initial metabolic changes described in macrophages is, of course, this oxidative burst. It's a very metabolically demanding process, which is directly involved in killing whatever the macrophage takes up. So yeah, I don't know. There's probably a mix of answers there in terms of mechanistically facilitating what it needs to do in order to kill pathogens, or on the other side maybe to resolve inflammation, as well as just on a bigger scale.
Well, yeah, I mean that's why I asked, because I looked at macrophages in fruit flies, and towards the end of the PhD, I became quite interested in metabolism, and we tested some stuff out, and it looks like actually activated macrophages in the fruit fly also go a bit glycolytic. So it's one of those things that I think has been shown in fish now. So it does actually kind of stick throughout evolution. So it's obviously a very conserved and important fundamental mechanism. The other thing I found, my last postdoc was working on CD8 T cells and hypoxia. And now one of the things we're trying to do is to sort of separate out hypoxia and metabolism. And
it's
really,
quite
really difficult to do. Like, what comes first? Because who knows? But yeah, I just couldn't quite figure out why they do it. I think it probably is, as you said, it's probably multiple reasons why they need to do it.
Yeah, I mean, my interest has always been kind of, on the other side, right? Like the alternative activation, type 2 immunity regulation, because my background is more understanding the immune responses to parasitic Helminth infection. And there's some really cool hypotheses about just, how your body metabolism changes in response to that. And whether, there are some papers that have kind of addressed this in a way, like whether that affects your immune compartment. And just also, I also like this idea of metabolic competition between the parasites. There's a cool paper from I think it was like early 2000s or late 2000s or something, where looking at the Helminth metabolism, and it's really dependent on fatty acid oxidation and lipids, as are supposedly alternatively activated macrophages, and some of the top secreted molecules by like the model Helminth that we often work with, Heligmosimoides polygyrus, are these molecules that act as lipid sinks. They just the proteins go and they have lipid binding pockets. They're just lipid scavengers. So I don't know. It's a hard question to tackle, but it's fun to think about.
Well, the thing, I think one of the things that surprised me the most was that a lot of the metabolites from switching their metabolism actually have immune function themselves. So, the big one would be itaconate that's what Luke kind of focuses on. But it's just really surprising to see these different roles for things that for such a long time we thought were we thought we'd understood something.
Seeing that a lot with lactate now, right? It's a waste product. No, it's not. It actually signals. It's I have to catch up on my reading on this, but on all these things. But it is. I agree with you. It's and honestly, I think that's one of the reasons that metabolism is such a big field. Is you pick your metabolite, you pick your molecule, your stimulus, or your protein of interest. It's almost guaranteed to have some sort of metabolic interaction or consequence or something.
How do they differ in metabolism from T cells?
Oh, well, they're way more metabolic in general. So, I mean, it makes sense in a way. They're much bigger cells. I think they're generally much more active in the tissues. Yeah, in terms of, different pathways, yeah, I don't know.
I mean, my understanding was they were generally activated T cells are slightly more glycolytic anyway.
Yeah, for sure. That's true. I mean, you see, similar changes in activated T cells as you do when you stimulate macrophages. But yeah, I think it's always hard to know whether something is going up specifically or whether it's just everything just becomes a bit more metabolic because of as the cell becomes bigger and more demanding.
Well, it's a question of what comes first, right? Is it metabolism driving these changes, or is it these changes then requiring more metabolism?
Yeah, it's for sure a bit of a chicken and egg. But I mean, there are parallels, I think, in within larger immune responses, right? So in a viral infection or something, or in my PhD I was comparing toxo infection, which is a very strong pro-inflammatory response, with Helminth infection, which induces these type 2 regulatory responses. And yeah, you see similar trends. Like cells involved in the type 2 immune side of things will generally seem to really have a preference for lipids, or at least metabolically they seem to take up and metabolize lipids more. Whereas you see this really strong increase in the glucose metabolism on the other side. So there's definitely, I think, parallels depending on the type of immune response you want.
And then, so you mentioned the M1, M2 spectrum. It's not M1, M2. M1, M2 is really handy, but it really is a spectrum. It's not nearly as... I wish it was that simple.
Yeah.
So in terms of, presumably, the metabolism is also on a spectrum. It's not all glycolysis or all TCA. It's probably on a spectrum. Well, how well does that spectrum map towards sort of pro or anti-inflammatory functions? Is it quite clear? Is there a definitive switch, I guess, is what I'm asking.
Yeah, well, in vitro, I would say, you can pretty clearly, define the metabolism based on the stimulus, or vice versa. It's really hard to say, to be honest. I mean, papers, it's such a complicated field. You tend to hone in on one pathway at a time and see whether it's playing a role or not. But it's really hard. I mean, I think these things are going to be hard to define.
I mean, we're here to ask the hard questions.
Yeah. I was just going to say, one thing that I think is cool about the field at the moment is there has been so much groundwork laid. So people have done these things where, you stimulate and you look at how metabolism changes. And what's cool now, and this is kind of what led my PhD, is now you can take a metabolic signature, roughly, and say, well, this fits with what has been shown before, and almost hypothesize that it's some sort of imprint based on that signal. And then, of course, you can test that hypothesis and see. And so, I think that is what's cool about metabolism now, and what we will hopefully be able to continue to do, where if you, instead of looking at what changes metabolism, look at the metabolism and use that as to try and infer now what is happening to the cell.
That's actually what I've tried to do recently in my current project.
Yeah, I mean, it depends. I mean, as you get more complex, it may or may not work. But I mean, in my PhD, we just kind of saw we have this signature of, a switch towards lipid metabolism. And, luckily a lot of immunometabolism has been done mostly with T cells. So, based off of, papers at the time, we made the hypothesis, and it ended up, I think, in a very cool finding, which will hopefully be published sometime. Unfortunately, not yet. But so that's my take.
So how does the metabolism differ across tissues? Because the general consensus is that within a tissue, a macrophage is sort of the M2, air quotes on a podcast. They work great. There's sort of the anti-inflammatory pro-healing type phenotype going on in tissues. So how do you see differences across tissues in terms of the macrophage metabolism?
Well, for sure. I mean, that's, I think, figure 2 of our preprint. Quite clear differences, actually. But, I mean, it's not just across tissues, even within tissues. And, I mean, I don't think we're necessarily the first to really think that this is the case. But I think our paper does kind of show this really nicely, because I can't think of any papers I know of. So sorry if there is one out there, but that have really done this kind of tissue-wide comparison outside of, say, single-cell RNA sequencing. And I'm not an expert in single-cell, but from talking to people who do it and from limited experience, it's really hard to detect metabolic enzymes and transporters because their transcripts are usually much lower, and you only pick up the top 500 or a few thousand or whatever, depending on what your budget is. But so I think that's where this the metabolic flow cytometry or spectral flow cytometry or CyTOF, whichever you're doing, works really nicely because it's you get nice single cell data.
At the protein level, of course, too. So it's actually probably happening rather than just transcripts, which may or may not be doing anything.
Yeah, I mean, of course, you run into other caveats. You can't say anything about the activity of that enzyme or that transporter. But we're working on trying to expand this panel or look at make kind of pathway specific panels. And hope that if you see that all enzymes are up in a pathway, you can make a pretty good guess that that's important. And then of course there are other ways we can work on validating this as well. But to just answer your question generally, I would say that the metabolic properties of macrophages vary dramatically across tissues.
So is this submitted now then?
Yeah, so we didn't get it through in our first choice, but that was also we knew it was a long shot, and it actually we ended up with more hope than we should have. But no, it's in review now, and we also have, we were invited to submit it straight to peer review in another journal that we will go to next, depending on the outcome.
Yeah, it's good to have options.
Yeah.
So, did you preprint at the point of submitting to the journal, or did you preprint before that?
Pretty much parallel to submission. So I think I put it on bioRxiv a few days before, and then my PI ended up doing the actual submission to the journal, and he did that a few days later.
It's probably good. That's painful.
Yeah, but no, I think there's a lot of discussions about preprints. I don't know if that's your next one of your questions.
It's probably one of my questions. The question we ask everyone is, whose decision was it to preprint? Was it yours? Was it the PI's, or was it a collective thing?
Collective. I think I was the first to say, I think it would be great if we could just put this out as a preprint, and he was just fully behind that. I think there are other people in our department who have also already put up preprints, so it wasn't new to, at least to my PI. But it's great to get that sense of just getting it out there, right? Because you never know how long the review process is going to take, and
long.
I've had colleagues who just get so sick of their papers by the end of it, which is such a shame because they put so much work into it. I feel like at least with a preprint, you almost get that instant gratification, even if it hasn't gone through peer review. It's it's out there for people to see, and it feels like an accomplishment, even if it hasn't gone through the whole rigmarole.
I've always been more excited at the preprint stage than I have at the actual publication stage, because I think it's that it's done, it's ready, here it is, and we can be all happy and excited about it. And then the whole peer review thing is, we've got to satisfy these people who may or may not have good opinions. And then, it's out, but it was already out. So why did you decide to preprint and not just straight up submit to a journal? Is there any specific reasoning behind that?
Some of it was for CV. Yeah, there were some grants that I was maybe going to apply for, which I didn't in the end, because it just didn't seem like it was wise based off of where things were. But so some of it was CV building, some of it's what I said. It's just it's just cool to have it out there. There's always a fear of kind of being scooped, and at least if you could put it on bioRxiv, you can I don't know if that would stimulate your competition to hurry up, or
well, I mean, if it's on a preprint server, it's out, so it doesn't matter if they hurry up.
But my hope would be if there's people working on similar stuff that they get in touch, and then we work on it.
We did that. We did that on my last
together.
Yeah. So my last paper, we preprinted it. A day later, a group in the US preprinted a similar paper, and we ended up working together and co-submitting. And they paid our APC fee as well for the journal.
There you go. That's an ideal situation.
Because we were, it was led by me, so we postdoc don't got no money for publishing. So the plan was actually not to publish; it was just to leave it as a preprint. So we were only able to publish because of that. So it was nice.
Oh, nice. Yeah, see, there are good stories in there.
There's lots of good stories. Or at least we get all the good stories. We've not had a bad story yet. Maybe we should hunt someone down who has a bad story to balance. do the PVC. So you also moved your lab, as you said at the start. So you were in Canada, then you ended up in Scotland. So how was that? Because it's stressful at any point during a PhD, I think, but moving labs is particularly stressful.
Yeah, I mean, I was more excited than anything. I think, academia aside, I had grown up in Vancouver all my life, and by no means a bad place to live, but I did have a sense of like I really needed to get out and experience something else, and this happened serendipitously. But yeah, I mean, a lot of time was lost for sure. I mean, trying to get things set up, figuring out all the new, admin and all of this, getting a new animal license.
Getting used to the UK very, very strict animal laws.
Oh, come to the Netherlands. Yeah, no, I keep going from stricter to stricter places. Yeah, Canada is... they still were, I think, okay ethically, but they made it just significantly easier in order to get things done. And the UK was a bit more strict, I guess, in a way. But ultimately you could still move forward pretty quick. The Netherlands is you have to like predefine your entire research before you can even touch an animal. And then if your project doesn't go the way that you planned, you can't. You have to go through a whole new application process to be able to address the new hypothesis.
How does that differ from the UK? So the UK license, you've got to do that as well. You're not really allowed to differ. You can request like changes to it. We've got kind of set.
Well, I don't know. I never had to write one, but my my experience was that you can write your license quite broadly.
Yeah, you can.
And as long as the procedure is on the license, then the actual kind of outcome or question that you're trying to address doesn't really affect it at all. But here, you have to kind of define your animal numbers for a particular experiment. You have these like no or no go moments. Where you have to say if this is we're going to do this experiment, and if this is the result, then we can do this experiment. If not, then we stop. That's it. But it doesn't really leave any room for like we did this experiment, it didn't work, but we found this interesting finding that we want to follow up on. And then if you want to do a different model or a treatment, or you want a new strain of mouse or something, it's like, okay, back to the application process.
Yeah, which is painful. I didn't realize it was that. Yeah, it's a weird way to do it, actually.
Yeah. So, anyways,
works.
I forget what. Oh yeah, moving labs.
Moving labs. That was the question.
Yeah. Yeah. No, it was a good experience. Definitely. I think I lost probably a year of like real productivity, but I mean, the advantages far outweighed anything that hindered me in that time.
Sunny Scotland. I mean, what more can you ask for?
No. Yeah, Scotland gets a lot of flack, but I love Scotland.
Scotland is a beautiful,
it's such an amazing
country.
place. Yeah, and Glasgow as well. I mean, also it still seems to have a bit of a reputation, but it was such an amazing city to live in.
There's definitely parts that are really, really nice. It's been a long time since I was in Glasgow, but like the whole of Scotland is an amazing country. Everyone should go to Scotland, and the Scottish Tourist Board should maybe give us some money for saying that. So we asked kind of what people are excited about and stuff, and your answer was new technology. So do you have a favorite new technology?
Honestly, no. I say that, but my bread and butter is like standard flow cytometry. But I'm starting to get into this. We submitted our first samples for single cell RNA-seq, so that'll be a fun challenge to wrap my head around. But I was just recently at a conference and looking at all the things people are doing. You can now do kind of all this in vivo tracing. What is it, like metabolic imaging, and just it's insane what we can now do. Of course, there's going to be financial barriers to most labs, or just where these machines are available. And there's someone associated with our group who's kind of trying to do some of these technologies, and it takes years just to optimize and get it to work. So I look forward to when people like me can just click a button and make these things happen. But
I mean, the metabolism field is probably a good field to be into and immune sort of cells, because those are two fields that I think are rapidly advancing in terms of technologies, or at least application of technologies.
Yeah.
So you've probably chosen the right place, or at least the right field.
Yeah, we'll see. I like it, so I'm here to stay.
Well, that's what matters. It's the important part. So, I mean, that leads us quite nicely to our other question, which is the other thing we ask sort of everyone is what do you think could be improved in academia? And I think you might be the first person to give the answer you give, actually. Now, so you said you had a really good experience in academia. So maybe we should talk about that, because we often, what we do with this question is we talk about what's wrong with academia. And of course, I mean, I talk about what's wrong with academia all the time because it's one of the things I research. But academia is really good. There's a lot of really good things. So this might be a really good opportunity for you to sell academia. All on you.
I mean, I don't want to come across as disillusioned. Like, I know there's several issues in your experience, some of them myself. But I read that question and just felt like, this seems unfair because,
it's definitely leading. It's a very leading question.
Yeah, but I mean, the one thing that this topic comes up semi-frequently, and the one thing I tell everyone, because at least this is my stance, is it really feels like a privilege to be in academia. I mean, from my side, I just I didn't really know what I wanted to do growing up, whatever. I stumbled into academia, and at the end of the day, it just, you get to play around, you get to just you're trying to figure out how things work, and it just feels like a hobby sometimes. And, I very much appreciate getting paid for my hobby, and I hope that no one will hear this and say, well, you're willing to work for free. But,
I mean, we basically work for free a lot of the time anyway. Don't worry, we've got the negatives covered.
But I mean, I don't know. It just, I think people maybe, don't think about that or forget that, or maybe they just don't feel the same way. But, it's just such a unique profession, I think, you have to. I guess you have to love it to stay the course.
I think that's definitely true.
And I think people that I often see disheartened or kind of decide academia is not for them is they just they don't really. It's not that they don't like what they do, of course, but they just don't have that kind of same feeling of like I'm getting paid to do a hobby.
I'm trying to think. See, I'm one of those disillusioned people. I'm trying to think now. So I'm in the process of trying to leave academia. So it's quite the feelings with that, because I don't want to leave. Like I was one of those people who knew what I wanted to do for like forever. Change a little bit, but once I knew it, I could stick for quite a while. And it was always have my own lab. It was always to be a PI and to have that, because like you said, it's you get to just explore, and you can find something and say, well, that's interesting, and then you can go away and do it, or you can write money to go away and do it. And it's quite a difficult thing to separate from, because I think science is very much you can do it 9 to 5. I know people who do it 9 to 5, but it is quite difficult to do that. You do need to kind of, because I guess with pay to think, right? So, you think all the time. My best ideas come to me when I'm trying to go to sleep, which is fantastic. But it's very difficult to do. And I don't want to leave it, but I think for me, it's great getting paid for it, but I think I feel like I should be paid more, or that I should have more free time. And I think that's where it gets me, I think.
Yeah, the free time issue for sure gets to me sometimes. It's it depends on on what's going on. I guess it's I think I'm someone that when I'm working on my own project and I'm into it, I will always work more than necessary. But the times that really gets to me is when you're directed.
Right.
And it's like, well, now I have to do this on top of what I would normally do, and then it gets a bit much. So, but that doesn't happen all too often. I've been lucky in that sense, I guess. But I don't know. I just I think it's just a unique career, and the flexibility it gives. I mean, you get opportunities to go to conferences and this kind of thing. It's stressful for sure. I mean, it's just I've also been lucky to have such nice colleagues, but I mean, it's always such support, such good conversation, scientific and non-scientific. There's always a feeling of kind of camaraderie.
I mean, that's a really important point, because I think that's the thing that makes or breaks academia a lot of the time, because all the things that are wrong with it, that's not going to change anytime soon. But the environment that you're in makes a huge difference. And if you're in an environment where you like people, well, not really like people, where you get along with people and everyone are happy to help out and chat and be social, that's a huge difference.
Yeah, absolutely, for sure. Yeah, I think it is one of the reasons I was so, moving to Glasgow worked out so well for me, is because the Institute in Glasgow really did this well. I mean, I was lucky to be with such a great bunch of PhD students and postdocs at the time I was there. But we had this big shared office of PhDs from different labs. We had our tea breaks together. We were all interested in each other's science. And went for drinks at the pub, of course, together.
I mean, you can't be in the UK and not do that almost every night.
But in Vancouver, I think the Institute just wasn't built in the same way, so it just came across as very isolated. So you really had to make the effort to try and find people to discuss things and get feedback, or just, go for a drink and have a chat. At least when I was there. I don't know if it's changed since, but I don't know. I like what I do.
So are you planning to stay then? Is the plan to open your own lab?
That is the plan down the line. I mean, we'll see. I've got a long way to go, of course. We'll see. See what obstacles come and if I can get over them. I will be at the stage, I guess probably sooner than I think, where I'll have to start applying for my own money, even just as a senior postdoc. Yeah, so we'll see. I find it difficult. That's one of the things I find difficult, actually, is, publishing, right? It's always kind of, at the end of the day, up to your supervisor to publish. And, if I can't get some of my publications out, in the next couple of years, then that really starts to, impact how I can do moving forward. I think I'll be okay. I mean, I've got papers in the pipeline.
Yeah, and you've got a PI who's happy to preprint, so you can skip that sort of 6-month wait. Because certainly, at least in the EU, most of the funding bodies now they count preprints as outputs. So that's all you need, really.
Yeah. I mean, I think it is, I keep hearing that it's changing. You don't necessarily need your CNS paper to move forward in your career. But we'll see when the time comes if that's true or not.
I think they still help, but there's a nice shift going on, which is good to see, because that's what this show's all about. But I think that was it. That's all my questions, I think. So unless there's anything you want to add, we'll fade out to music. John can fade out to music.
Yeah, if we had whiskies, I would cheers you and just say have a nice night. But...
Mine's gone. It's empty.
Okay, and that is the show. If you enjoyed listening, then hit that subscribe button for more and leave us a review on whatever platform it is you're listening on. You can reach out to us on Twitter at MotionPod or online at preprintsandmotion.com. Didn't enjoy that? Well, we're all scientists here, so send us your review and let us know what works or what you'd like to hear more of, or less of. But until next time, have a good week.
Where do I find out about the different bioarchive licenses? This CC, BY, CDXY nonsense is driving me nuts.
ASAP Bio have a resource for that.
Ugh, that's your answer for everything.
That's because they have everything you need to know about preprints.
Sure, they probably have the basics, like info on the preprint servers, but what else is there?
There's so much more. Looking to post a preprint, but not sure what different journal policies are? They have a collection to help you out with that. There are meetings around preprints and associated services. If you want to know how preprint adoption has changed over time, there's even a page on that.
And COVID?
They have a big section on preprints and the pandemic, plus some really cool infographics for communicating preprints.
And university policies? Surely they don't have that.
They collect uni policies where possible.
Okay, they do sound pretty impressive, but is it not a bit of an echo chamber?
It can be, but ASAP Bio also engage with people who don't love preprints and have concerns. So we had an excellent discussion on this very topic a couple of months ago.
Oh, is there anything ASAP Bio don't do?
Honestly, no, they're so nice over there. They were so quick to jump in and support this show. It's your one-stop shop for info on preprints and open science initiatives, so head over to asapbio.org to learn more and subscribe to their newsletter for the latest in preprint news. If you want a deeper dive into the world of preprints, then look out for the next recruitment of ASAP Bio Fellows.